viruses. Okay, and viruses have been in the news, of course, a lot lately with COVID-19 and all the efforts to try and control the viral spread. Right, a virus is not a living thing. Okay, it flunks two of the criteria to be a living thing. Okay, it is not cellular. Okay, not cellular in structure. Okay, and it can't reproduce on its own. Okay. The genome is either DNA or RNA, but never both at the same time. And it can be linear or circular if it's DNA, and it's either one linear molecule or several linear molecules if it's RNA. Okay. Um, it needs a host to reproduce. So it's an obligate intracellular parasite. And there's a virus or something similar to a virus for every living thing on the planet. Okay. Taxonomic classification is difficult. Currently, viruses can be classed based on shape, nucleic acid type, mode of replication, host, and the disease they cause. International Committee on the Taxonomy of Viruses puts them in the order Virales, the family Viridae, subfamily Virinae, and the genus virus and the species virus. Okay, that's this. Okay, so there we go. Okay. Classification of virus is tough because of the lack of fossil record and the ongoing debate on non-living organisms. The ICTV is responsible for the development of the current classification system. In the early 90s, it devised and implemented rules for naming and classing viruses. This system shares features with the classification system of living things, such as taxon structure. On the basis of shared properties, the viruses are grouped at the hierarchic levels of order, family, genus, and species. The Latinized suffixes are shown in italics within the taxon. For the determination of order, the type of nucleic acid is indicated. The presence or absence of an envelope is used. In addition, the other characteristics, such as the type of host, the capsid shape, and immunological properties can be included, as well as the type of diseases that they cause. Okay. So you can see here with the Baltimore classification, places the virus into one of 7 groups, distinguishing the viruses on the basis of the relationship between the viral genome and the messenger RNA. So you can see the groups are just Roman numeraled 1 through 7. Okay. The shape can vary from very small, about 20 nanometers in parvo, to up to 450 nanometers, is also almost half a micron in poxvirus. You can't see them with light microscopy. You have to use an electron microscope. Okay. And that didn't come around until much, much later, which is why we knew relatively early after the invention of the regular microscope that there were things smaller than a cell that could cause disease, because we could filtrate from a diseased plant, for instance, through a porcelain filter, which has pores too small for a cell to get through. And the filtrate could cause disease. So we knew something tiny, tiny, tiny could produce disease. The electron microscope came along decades later. Okay. The exception to size is the Mimi virus, found in 2003 with a diameter of almost a micron in free-living amoebas. It's the largest known viral genome with 1,000 genes on a double-stranded circular RNA. So you can see some size comparisons there, right? The size of a bacterial cell is shown below, right? And then you've got these viruses, right? And you can see the relative size. So this is one of the reasons why the masks didn't work in stemming the spread of COVID. Okay. The virus is too small and it will get through a typical paper mask. All right. It's sort of like trying to say that you could use a colander to hold water. You can't. Okay, the water goes through the holes. An N95 can do it. Those holes are very small, but those masks are expensive and you have to wear them all day, okay, for them to be effective. So it's just a sort of word of warning, okay? You have to understand what you're actually dealing with and look at the actual research in order to form your opinions and your behavior accurately, okay? Just look how tiny these things are, okay? Viruses are basically genetic material, and they're enclosed inside what's called a capsid, all right? And the capsid is made up of interlocking pieces called capsomeres, all right? And And basically, when a virus invades a host, it turns it into a virus factory. And the result then is that you take the cell off task, and that can produce disease. And you can also produce cytopathic effects inside the cell. Okay. Some viruses are enveloped, meaning that they have a plasma membrane or a nuclear membrane around them as they leave the host as a way to disguise them from the immune system. Okay. The capsomere and the capsid is the complex structure that serves as the basis for morphological distinction. Okay, so you can see an example of a typical animal virus here. The spikes are important because the spikes are one of the ways that a virus can dock and penetrate a host cell. It's like a key that unlocks a lock, right? The spike engages a receptor and then receptor-mediated endocytosis pulls you in. Okay, so you can see the capsomere, you can see, in this case, they're showing you some envelope, right? That's the green stuff, and the spikes, of course, outside. Proteins associated with nucleic acids are called nucleoproteins. The association of viral capsid proteins with viral nucleic acid is nucleocapsid. Spikes are long projections from the nucleocapsid, and the virion is a fully assembled virus. According to the shape and arrangement of capsomeres in the presence of an envelope, viruses can be split into helical, isocahedral, enveloped, and complex viruses. So let's look at each of these, right? Helical viruses are capsids with rod-shaped capsomeres. The genetic material can be single-stranded RNA or single-stranded DNA, and it can be naked, such as in tobacco mosaic virus, or it can have an envelope, such as in flu virus. Helical capsids are going to have a long axis resembling a wide ribbon stacked around a central axis, forming a helical structure with a central cavity like a hollow tube. As a result, these virions are rod-shaped and filamentous and can be short and highly rigid, as in plant viruses, or long and flexible, as in animal viruses. The picture shows you the structure of a TMV. The capsomeres of the naked helical virus are rigid and tightly wound into cylinder-shaped structures. Okay. Icosahedrals are three-dimensional geometric figures with 12 corners, 25 triangular faces, and 30 edges. The arrangement of capsomeres varies between the viruses. Examples include herpes viridae, adenoviridae, papoviridae, and parvoviridae. Okay. During assembly of an icosahedral virus, the nucleic acid is packed into the center, forming a nucleocapsid. Icosahedrals can be naked, such as adeno, or enveloped, such as herpes. The figure shows an icosahedral. The capsids of these viruses are called icosahedrons. They're three-dimensional geometric figures with 12 corners, 20 triangular faces, and 30 edges. What you notice about all this is it looks nothing like a cell, okay? And that's why it flunks the criteria for life based on that alone, okay? An enveloped virus surrounds the nucleocapsid, is typically obtained by budding through the host membrane or the nucleus, and steals the envelope as a way to disguise it from the immune system. It might include some viral glycoproteins embedded in it as well. Some or all host membrane proteins are replaced by viral proteins. The viral envelope created through the budding process does not disrupt the plasma membrane and doesn't kill the cell. So some budding viruses can set up persistent infection. The figure shows a flu virus, which is an example of an envelope virus with two different types of spikes, hemagglutinin and neuraminidase, H and N. A bacteriophage is similar to a virus, except that its target is bacteria. Okay? It has a head and a capsid with nucleic acid in it, varying in size and shape, made up of one or more proteins. The tail is a hollow tube surrounded by a sheath and a base plate with attached fibers. So the idea is this thing lands on the bacterial cell and injects the genetic material like a hypodermic syringe. These viruses are complex. They consist of a capsid that is neither purely helical nor completely icosahedral, and has extra structures such as protein tails and a complex outer wall. The pox viruses are large, have complex DNA viruses with an unusual morphology. Okay, they're called bacteriophages because that means bacteria eating, basically. An even more complex group of viruses are bacteriophage, viruses that infect bacteria in general. It has an icosahedral head bound to a polyhedral tail attached to a base plate with a lot of protruding protein tail fibers. And the figure shows the structure of bacteriophage. Although different in size and shape, they all have a capsid that contains the genetic material. A lot of bacteriophages also have a tail that enables the bacteriophage to attach to the host and inject the genetic material. Right? Pox viruses lack a regular capsid. They have a nucleoid region surrounded by membrane and two lateral bodies. They're about 200 to 300 nanometers long and have a complex nucleocapsid symmetry. They vary in shape depending on the species, but for the most part, they're brick-shaped or oval and similar to a rounded brick. Okay. The figure shows a vaccinia virus, member of the Poxviridae family. The genetic material can be DNA or RNA, but never both at the same time, okay? It can be double-stranded or single-stranded DNA, double-stranded or single-stranded RNA. It can be either linear or it can be circular. Generally, species will contain one or the other. One exception is cytomegalovirus, okay? Reverse transcription or retroviruses have an RNA genome but produce a DNA copy through the action of reverse transcriptase, which is a unique enzyme and presents a drug target for antivirals for that class of viruses. The size of the genome varies substantially among these different species. That DNA copy of the retrovirus integrates into the host genetic material. All DNA viruses, as well as these retroviruses, perform some of their life cycle in the nucleus of the host, and that makes them extremely difficult to get rid of, okay? In some cases, if you're infected with these kind of viruses, you have them forever, okay? RNA viruses are different, right? RNA viruses do all of their work in the cytoplasm, and the body has mechanisms for clearing them. And we also have some antivirals that are effective against them, okay? So let's talk a little bit more about viral replication. Viruses are acellular, so they don't replicate through cell division. They use the host to assemble and release them, okay? Outside the host, the virus is simply an active or an inactive particle. Bacteriophage are often used in diagnostic laboratories for the ID of pathogenic bacteria through phage typing. Each phage is very specific for a particular bacterium. Phage contain either DNA or RNA. Their attack cycle includes absorption, penetration, replication, assembly, maturation, and release. Bacteriophages are common in all natural environments, and their presence is related to the numbers of bacteria present. The phage life cycle is important to understand so that we can understand the mechanism by which bacterial genes can be transferred from one bacteria to another. The events that occur in the multiplication cycle of bacteriophage, listed here, include adsorption, which represents the recognition process between phage and bacteria, followed by attachment, and then penetration of the phage DNA into the bacteria. Replication represents copying of the phage nucleic acid, followed by the assembly of the phage parts and the maturation of the newly formed phage. The mature phages are then released from the host cell and are ready to infect others. So the idea here, right, is that we land, we penetrate, and then we take over. The minute the genetic material is free, then we are ready to begin transcription, translation, and assembly, okay? And the normal function of the cell is gone, okay? In adsorption, the phage has to find a susceptible host attached to the bacterial cell. It's mediated by tail fibers or analogous structures on phage without tail fibers. Phage receptors on the bacterial surface include proteins on the cell wall or pili and flagella. The host specificity of phage is commonly determined by the type of tail fibers on the phage. The first step is attachment of the bacteriophage to the bacterium by the tail fibers, which is weak and reversible. The second step is irreversible binding of the phage to the bacteria, mediated by components of the base plate, or alternative ways by other bacteria that don't have this. Firm attachment to this bacteria is irreversible. The phage is now positioned for penetration. Okay. In penetration, after the irreversible binding, we inject the genetic material into the bacteria, just like a hypodermic needle. The contractile sheet constricts after absorption and pushes the inner tube through the cell wall of the bacteria and the cell membrane. Generally, only the nucleic acid enters the cell while the phage stays outside. Okay. Penetration by some bacteriophages that attach to pili rather than to the cell wall results in both genomic DNA and capsid entering the cytoplasm of the host or the recipient. Okay. All right. Let's pause. Well, let's do this one, right? In replication, bacterial metabolism shifts to gene expression and viral nucleic acid production, and then translation, right? Of viral protein. The result is production of viral particles, right? Proteins that seal the damaged bacterial cell wall prevent premature bacterial death. Enzymes are necessary to copy the genome of the bacteriophage. They can be host or they can be coded by the virus. Proteins are needed for the formation of the capsid head and part of the tail fibers, okay? Enzymes are required for the weakening of the bacterial cell wall to lyse the bacterial cell, if that's what this phage does, and release the newly assembled bacteriophages, okay? Now, some phages don't blow the cell up when they reproduce. Some of them either go into what's called lysogeny, okay, where they sleep with their genetic material integrated in the bacterial chromosome, or they can actually extrude the virus through the membrane of the host cell. And in that case, the bacteria doesn't really die, it just grows very slowly. An example of that is M13. Okay. Let's take a little 15-minute break. Yeah. Okay, so replication is just the production of new viral particles. The result is the ability to spread their range, right? This change in metabolic activity of the host cell results in the formation of viral molecules, including proteins that seal the damaged bacterial cell wall and prevent premature bacterial cell death. Enzymes necessary to copy the genome of the bacteriophage, proteins for the formation of the capsid head and the tail fibers, enzymes for the weakening of the cell wall to lyse the bacterial cell and release the newly assembled phage, right? With a lytic phage, all right, also called a virulent phage, after multiplication, they blow the bacteria up and the phage is released into the environment. In the eclipse period, no infectious phage particles are found in the host cell. Intercellular accumulation phase is assembly. Lysis and release is where we explode, okay? So you can see here how that works, right? Now, you might well ask the question, okay, is it always a good idea to blow up your host? And the answer is no, right? If these particles blow up their host and there's no other nearby host to attach to, you've sort of limited your ability to spread your range. Okay, you've blown it. So generally speaking, if they can do it, right, these bacteriophages can alternate between what we're going to look at next, which is the lysogenic cycle and the lytic cycle, okay, in which the lytic cycle, where they blow up the host cell, only happens where there's other nearby host cells. In other words, where the population density of bacteria is high. If there aren't a lot of nearby cells, then instead what we do is we integrate into the host genetic material and we wait for those conditions to appear, all right? And so as the bacterial cell reproduces, so does the genetic material from the phage. And that's what lysogenic phages are all about. Phage genes are in the host but are not transcribed. It's in a stage called prophage. The phage DNA is integrated into the host chromosome and passed on to the daughter cells of the bacteria. It might persist indefinitely or eventually lyse the cell. Okay, those phages are those that can either multiply by the lytic or undergo absorption and penetration into the bacterial host, but are not replicated or released at that time. So it's just a strategy for determining, when to do my thing. Now, with this lysogeny, where we integrate it to the host chromosome, sometimes when we go back into the lytic cycle, we can accidentally take some of the host genetic material with us, all right? And that gets loaded into a particle. And if that particle infects another host cell and lysogenizes it, it can give it a gene for a virulence factor, a weapon to produce disease. And that's going to be the case in a lot of the bacteria that we look at that cause human disease, okay? This is one of the ways you can move genetic material around very quickly in bacterial cells, okay? Animal viruses are similar to the bacterial multiplication stages. The naming of the stages is the same, okay, except for uncoding, because what happens here is that the capsid actually gets inside the host cell, okay, but everything else is the same. Although the basic stages of multiplication of animal viruses are similar to phage, several differences are present between the host cells, their viruses, and also in some parts of the multiplication cycle. In absorption, we attach the virus to the host. The viruses are specific for a particular host and infect only one or a restricted range of species, and that's called their host range. Attachment sites of the virus are distributed over the capsid or the envelope, okay? Animal cells that lack receptors for a specific virus are resistant and can't be infected, right? Naked envelope viruses differ in the mode of attachment to the host. Naked viruses have surface molecules that adhere to the membrane receptor, whereas envelope viruses have glycoprotein spikes that bind to the plasma membrane receptors of the host cell, okay? But basically, you're tricking the cell into letting you in, all right, bottom line. Now, if you have a mutation in these receptors, you can't get inside, okay? And so you're resistant, all right? That can be present in the population naturally, and it can even be selected for by other types of diseases, okay? It's thought, for instance, that the descendants of the survivors of the Black Death in the Middle Ages. A lot of them inherited resistance to AIDS, okay? And as a result, they can carry the virus, but they don't get sick, but they can transmit it to other people, all right? And that's because of a variation in the CD4 receptor on the helper T cell. In penetration, the virus enters the cytoplasm of the host. Endocytosis of the whole virus, fusion of the viral envelope with the plasma membrane of the host occurs. Only the nucleocapsid enters the cell. Unlike bacteriophage, animal viruses generally don't inject their nucleic acid into the host. On occasion, non-envelope viruses leave their capsid outside the cell while the genome passes into it. In uncoating, viral nucleic acid gets released. Uncoating process varies between viruses. If virus is entered by receptor-mediated endocytosis, the virus is enclosed in a vacuole or vesicle, and enzymes within these dissolve the envelope in the capsid and release the genetic material. Another method is direct fusion of the viral envelope with the plasma membrane of the host, releasing the nucleocapsid into the cell cytoplasm. Some antiviral drugs exert their effect by preventing the uncoating of the nucleocapsid, okay? These are called fusion inhibitors, okay? Replication occurs immediately after uncoating. In DNA viruses, viral DNA gets into the nucleus of the host cell, is transcribed into message, which leaves the cytoplasm and is translated by host ribosomes. Early viral proteins help the replication of viral DNA. RNA viruses provide their own polymerase for transcription, so that provides a drug target, okay? Retroviruses use a different route. They use reverse transcriptase to make a DNA copy of their RNA, and that DNA copy then integrates into the host DNA. In all three cases, the translation of viral message occurs with host ribosome and produces viral proteins for the final assembly, right? So this phase is regulated and complex, and the events proceed depending on whether the nucleic acid of the infecting virus is DNA or RNA, okay? In addition to viral protein synthesis, the virus has to replicate its genetic material. In the early phase, the proteins produced are enzymes and regulatory molecules that allow subsequent replication of viral nucleic acid. In the late phase, we have synthesis of proteins needed to make the capsid, to build the box to put the genetic material in, okay? So a couple of things about this, right? There are going to be stages in some of these viral replication cycles in which there are vulnerable intermediates. Okay, so let's talk about that for a second. Okay, so you can have in the virus, okay, during replication, like double-stranded RNA in RNA viruses. Okay, and you can have the production of RNA from DNA in retroviruses. Okay. And that's mediated by something called reverse transcriptase, RT. Okay. Double-stranded RNA is not normal. Okay. We don't make it in a healthy cell. Okay. So this is a target. Okay. That's a target. Reverse transcriptase is not normal, okay? We don't make it, right? We don't make DNA from RNA, so that's another drug target. So the body has solutions for this, okay? And that's interferon and an enzyme called dicer, okay? Since these guys work exclusively in the cytoplasm. RNA from DNA, we can target reverse transcriptase with drugs like AZT. Okay. So these are antivirals. Okay. These are antivirals. These you can use after you've been infected. All right. And how about the viruses that just do DNA? Well, problem is you can't get rid of those if they hide inside your own tissue. If they integrate or if they hide in the nucleus, there's no way to get to them. Okay, so you got those for the rest of your life. An example of that would be herpes zoster, which causes chickenpox and shingles. If you get the zoster, it's going to migrate to your dorsal root ganglia, and it's going to live there basically forever. Okay, and it can reemerge as shingles later on. Okay, all the herpes viruses can do that. Okay, so generally, when we talk about avoiding a viral disease, we're talking about a vaccine. And a vaccine is only going to be as effective as the mutation rate of the target. Okay? If the target virus mutates at a high rate, a vaccine just really isn't going to help you much because you'll be resistant to what's in the vaccine, the antigens in the vaccine, but that may not be what's out there in the wild that you encounter, and you'll get sick. Okay? If the mutation rate is low, then the vaccine can be very effective. So two examples that come to mind are polio vaccine, which has been effective for many decades because the rate of mutation of polio virus is very low. All right. Whereas what we just saw with COVID is that the rate of mutation of the virus is very high. And so a vaccine for COVID is kind of pointless. Okay. It's kind of like trying to make a vaccine for a cold virus. All right. COVID basically is an engineered cold virus. And the result is that you'll be resistant to what's in the shot, but the likelihood that you're going to get a cold is very high. And the same thing that we've seen with COVID, right? You can get a COVID vaccine and turn around and get COVID. Why? Because it mutates. Okay? So that's the battle you're up against, right? In assembly and release, mature viral particles are made from a growing quantity of viral components. Two ways of release are budding or exocytosis and lysis, where you blow the host up, right? Once synthesis of viral components is complete, assembly begins. Commonly, the capsid is made first as an empty shell and is a container for the genetic material. This is called encapsidation. Although budding of envelope viruses causes them to shed gradually over a period of time without sudden cell death, most active viral infections are ultimately lethal to the cell because of damage caused by the virus. So you can see here with a naked virus what the steps are, okay? Pretty well outlined. Okay? And you can see with an envelope virus where we steal some of the host components like plasma membrane or nuclear membrane. Sometimes you even steal transfer RNAs, okay? And we're out. The envelope, of course, is a disguise so that the immune system cannot detect the pathogen, okay? Abortive viral infections don't have viral production. Lytic or cytocidal infections kill the host. Persistence are not lytic but productive. Latent results in limited synthesis with no viral synthesis. The virus remains asymptomatic for long periods of time, such as herpes zoster or herpes simplex, okay? Slow infections are prolonged incubation. Damage takes years, followed by disease. Transforming infections, the viral nucleic acid can remain indefinitely without virus production, and you can end up with possible cancer changes. So viruses are mutagens and they can cause cancer, okay? Short and long-term effects in animal cells are well documented. Lytic infections occur in permissive cells, which provide biosynthetic machinery to support complete replication cycle. Persistent infections don't cause cell death. The effects of chronic are mostly the same as acute cytocidal, with the exception that the production of the virus may be slower, intermittent, or limited to only a few cells. Although the progression of cellular damage from a slow infection can take years, it's followed by extensive cellular injury and disease. The term oncogenic transformation refers to genetic modification of the host to turn it into a cancer cell. Okay? What are these effects that damage the cell? Well, morphological or cytopathic effects include changes in shape, detachment from tissue surfaces, lysis, membrane fusion to form what are called syncytia, altered membrane permeability, inclusion bodies, and cell death. Physiological effects, addition of viral proteins into plasma membrane, change of characteristics such as changes in ion movement and other activities leading to changes in cellular activity. Biochemical effects, inhibition or alteration of host cell macromolecules. Genotoxic effects, genotoxic substances that damage host cell DNA, causing mutations, potentially resulting in cancer, right? Viral proteins incorporated into the plasma membrane of the host can change antigenic or immune properties and can cause autoimmunity. Cytopathic effects caused by different viruses depend on the virus and the cells they hit. The observation of cytopathic effects can be used in clinical virology to help figure out what virus you've got. Okay. Cellular transcription and protein-to-protein interaction can be changed to promote production of new virus. Some viruses stimulate specific cellular biochemical activity to increase virus replication. And then we talk about genotoxic substances, right? Major groups of viruses and vertebrates cause diseases after they break into the protective barrier. Most viruses are specific to a particular cell or tissue type. However, a particular disease can be caused by several viruses. The genome of viruses is either DNA or RNA. The disease can be caused after the viruses gain access to underlying tissues and organs. Example, adenovirus. There's 57 serotypes that have been ID'd in humans. Usually stable to chemical physical agents. There's a recent interest in adeno as vectors in gene therapy, and they cause a lot of respiratory illnesses. The virus is capable of infecting multiple organ systems. Most of these infections are asymptomatic. In children, adeno typically causes respiratory and GI tract infection. Immunocompromised patients are especially vulnerable and susceptible to severe complications of adeno. It can cause gastroenteritis, conjunctivitis, or cystitis. Herpes adenovirus, cause hepatitis in humans and animals. Examples include Hep B. It can cause chronic, symptomatic, or asymptomatic disease. These viruses are enveloped virions with a small circular genome with partially double-stranded and partially single-stranded DNA. When the virus enters the host cell, the single-stranded portion is restored to a double strand by a DNA polymer that's in the core. Okay. Herpes simplex 1 and 2, and varicella zoster. Okay. Papilloma and polyoma virus, which cause warts and possible cervical carcinomas. Okay. Parvo, B19 causes human disease. Others are animal pathogens. And pox viruses, which are the largest, cause smallpox. Okay. When herpes virus is activated, it can cause recurrent disease in their host. Reactivation of the virus can occur in response to environmental changes such as UV light, X-rays, cold, hormonal changes, stress, okay, and immune deficiency. HPV and polyomavirus can cause host-dependent lytic, chronic, latent, and transforming infections. They're small and non-enveloped with circular double-stranded DNA and an icosahedral capsid. Parvo has a non-enveloped icosahedral capsid, and the B19 viral genome contains a linear single-stranded DNA molecule. B19 replicates in mitotically active cells like human bone marrow, erythroid cells, and erythroid leukemia cells. It's an agent of contagious erythema infectiosum, commonly called fifth disease, because of the development of a face rash similar to the other 4 diseases that produce rash. The vaccinia virus is related to the smallpox virus. It's the live virus used in the smallpox vaccine. When given to humans as a vaccine, it will help the body develop immunity to smallpox. The vaccinia virus is best known for the eradication of smallpox announced by the WHO in 1980. Okay. Bunia viride is arthropod-borne. Okay, it's spread by an insect. Okay, coronavirus is a respiratory and enteric disease pathogen, cause of SARS. Okay, it's the most prevalent cause of the common cold, rhinoviruses being the first most common. Hep viruses cause hepatitis and cirrhosis and hepatocellular carcinoma. A lot of these liver diseases, which are classed as hepatitis, are viral, coming from a wide range of families. Viral hep is a major public health problem all over the world, affecting millions of people. Okay, orthomyxovirus, okay, includes flu A, B, and C. Type A is the most virulent. Flu viruses are respiratory viruses responsible for flu-like symptoms, including fever, malaise, headache, and body aches. Flu 1 is one of the most prevalent of the viral infections. The prophylactic vaccines and antivirals that are now available make it possible to prevent serious disease complications in people at risk. Morbillivirus includes the measles virus. Paramyxovirus includes parainfluenza and mumps. Pneumovirus includes respiratory syncytial virus, or RSV. Protection can be provided for the measles and mumps viruses by an effective live vaccine used in the US and other developed countries. RNA viruses. Picornavirus. Important pathogens include enterovirus, rhinovirus, hepatovirus, aphthovirus, earbovirus, corbovirus, and tessiovirus. It's the largest family and has the smallest RNA genome. Two categories causing a variety of human infections are entero and rhino. People not vaccinated and who have been bitten by an animal or otherwise been exposed to rabies can receive vaccination after exposure to prevent the disease. The name reovirus was given to a group of enteric and respiratory viruses not associated with a particular disease and were considered to be respiratory enteric orphans. Okay, so rotavirus example, gastroenteritis in children, over 50% of the case of diarrhea in children and requiring hospitalization. Okay, rotaviruses are one of the causes of travelers' diarrhea. Okay, rhabdoviruses hit plants, insects, fish, birds, mammals, and humans. Rabies belongs to this group. Okay. Retroviruses, lentivirinae, supinavirinae, example, HIV. Okay, they're classed according to the disease they cause, tissue tropism and host range, virion morphology and complexity of the genome. Alpha viruses are found around the world and many are pathogenic to humans. Okay. The most common results of alpha viral infections include infectious arthritis, encephalitis, rashes, and fever. Alpha viruses are of interest in gene therapy for the development of viral vectors of gene delivery. Okay. Flaviviruses are envelope viruses with single-stranded RNA and icosahedral nucleocapsid symmetry and make them similar to togavirus, but differ in their method of replication. Okay. All right, subviral agents I'm going to save for when we meet again tomorrow. And there are some interesting stories that surround this. This is the last slide in this chapter. Okay. So with that, I'm going to end here.